Okay, so when a couple decides to have children, we send the woman in for her prenatal medical visits. We offer her extensive counseling on the dangers of smoking, drug, and alcohol use. We even make sure that she's eating her vegetables and taking her prenatal vitamin. In contrast, the health of the father is dismissed as irrelevant. And so what I want to argue today is that that perspective has complicated our understanding of pediatric disease, and it has created a significant blind spot in developmental toxicology in general, especially as it relates to understanding FASDs, where the warning label on the bottle tells us where the problem comes from to the exclusion of all other possibilities. And so what I hope to do today is to convince you by showing you some biomedical evidence that male drinking is a plausible contributor to fetal alcohol spectrum disorders. And I want to talk about the role of the epididymis, a specific section of the male reproductive tract that we think are programming these effects. And then finally, I want to talk about the role of mitochondria and the role that we think that they are carrying out in executing these effects in the offspring. Okay, so here are the learning objectives for class today, and so just let me say this is probably the best attended 8 o'clock lecture I've ever given in my life, and I've been told I have 10 extra minutes, so man, the stars are aligning for me today. Okay, so what we're going to begin with is reviewing the role of paternal alcohol exposures on offspring health. And then this is going to lead us very logically into a conversation about epigenetics. And so I want to spend some time here defining this term and talk about how it is that a memory can persist across the life cycle. Then we're going to start talking about male reproductive physiology and discuss the role of paternal health in programming different epigenetic outcomes. And then I want to start talking about the role of paternal contributions to fetal alcohol spectrum disorders. And then bridging off of this, I want to start us to consider how maternal and paternal exposures might interact in driving FASDs. And then we're going to move into a discussion of mitochondria and their role in disease, and then start to consider evidence that FASDs might be a condition that could be unified or linked by considering bioenergetics. And so there's a researcher by the name of Doug Wallace who advocates that a lot of different ailments in society, ranging from Parkinson's disease, Alzheimer's, dementia, obesity, cardiovascular disease, all of these things could be unified if we started to think about mitochondria, and then he turns around and he chastises the geneticists for only considering 2 of the 3 genomes that we have. We each have a genome that we get in our mitochondrial genome that we inherit from our maternal grandmother, yet this is not something that's widely considered in genetic analysis. Okay, so what I study in broad terms is trying to understand how the memory of an exposure or a stress at one phase of our life cycle persists and has an effect later in life. And so this field of research was really born from the observations that different stressors, be them nutritional stressors, stressful encounters in the society or on the person, or exposures to different toxicants, had these effects, some of which manifested very early in life, but a large number of them actually didn't show up until later in life. And so we referred to this as the fetal origins of adult disease hypothesis. And so we've been able to correlate very nicely some exposures during fetal development with the onset of cardiovascular disease, diabetes, gut inflammation, and various pathologies in the liver. And so importantly, when we start looking at this, one of the things that can unify all of them is a dysfunction in the immune system. And so I want to plant this seed right now, and then I'm going to come back to it towards the end of my talk. However, as we started to study this phenomenon, we became aware that it was really epigenetic changes that were driving it. Well, then a lot of people in the field really started to question, why is it that we're only focusing on the mom? Why is it that we have this maternal-centric focus? Why are we ignoring dad? And so if you consider this, the reality is that males are more prone to abuse drugs, they're more prone to a sedentary lifestyle, there's more social pressure on women to look good, to look athletic and look fit than there is on the male partner. And then conversely, males are more apt to experiment with drugs and do just generally dumb and risky things. And so, each of the exposures that I have up here has a demonstrated impact on mitochondrial function and causes oxidative stress, which we know is a significant modifier of male fertility. And this is across a number of different species, not only in humans, but into the field of agriculture as well. And so the argument that I want to put forward today is that instead of just considering the outcomes associated with one parent, what we need to do in order to understand the developmental origins of pediatric disease and environmentally induced disease is to start considering inputs from both parents, all right? So there's the social determinants of disease here, and we have to consider both parents because both generally share the same environment, both are going to share the same stressors, and so it just makes sense to start looking at it from both angles. All right, and so the reason that we've been so hyper-focused on maternal exposures is that it's so much easier to draw a straight line between the exposure and the outcome. Mom drank alcohol, and here's the consequence to the fetus, right? There's a direct, straight line between these two things. And so to this point, most of developmental toxicology is looking for an immediate outcome. What is the poison doing at a particular time in development? However, we've started to recognize that in contrast to direct effects, and so what I mean by direct effects is that you have this phenomena where a developing embryo is exposed to some kind of stress, a poison, radiation, just, there's a number of different things that we can look at. And this then causes an immediate outcome where the cells die. And so this has kind of been the mindset in toxicology for the large majority of history because it just makes the most sense. However, we're starting to understand now that a lot of toxins, particularly alcohol, extend outside of the window of exposure. And so Carl Jung in his talk the other day talked about modifications to the operating system. And that analogy is spot on. And so what we think happens is that there's a change in the operating system of the cell where it starts to function a little bit differently. We haven't hurt it or killed it in the sense that we've thought about in the past, but what we've done nonetheless is to change fundamental aspects of the cellular physiology such that we've started to program a predisposition for disease. Okay, so I, like many of my scientists, brothers and sisters in this room, have lost the ability to speak to regular people. And so to all of the persons with FASD, to their parents, to their caregivers, and to the people who are supporting them, I want you to know I'm aware of the problem, I'm working on it, and so going forward, I've put a statement at the bottom of the slide, what does this mean? Okay, and so as I stand up here and I use my technical gobbledygook to describe what I'm doing, if at any point you get lost here, please look to this summary down at the bottom of the screen, which I hope provides a very plain description of what it is I'm actually trying to say. Okay. And so this then brings us to a discussion of epigenetics. And so in essence, what we're trying to do here is to understand how is it that the cell retains a memory of this past exposure. And so in essence, what epigenetics is trying to understand how the information is organized within the nucleus. Okay, so we get information in the form of genetic information from our parents. How is that information organized inside the nucleus? And so the analogy that works best here is to start thinking of this as epigenetics as being like a book. If the pages of the book are open and you can see the information, you can read it, okay? And if the pages are shut, you can't see it. And so what happens here during development is that various regions of our genome get organized into loops that are in this wide open conformation which can be read, or in contrast, put over here where the information is scrunched up and now silenced. And so what happens through development is that each cell has a unique way in which it starts to package the genome. Certain genes get pushed into the open conformation, and certain genes get pushed into the silent conformation. And so as an example, a pancreas cell, a pancreatic beta cell, is going to take the information for insulin, which it needs, and it's going to put it in an open conformation because that's a fundamental component of its cellular physiology. That is its job. In contrast, it's going to take the gene for hemoglobin, which is only expressed in red blood cells, and it's going to take that genetic information and put it over here. And so this is the way that through development, we go through these periods where we start to reorganize our genetic information, and we are going to partition it into regions that we can see or not see, just depending upon the cell type. And this is a fundamental element of cellular identity. And so what we're trying to understand here with developmental programming is how do inputs from the environment change this organization? All right, and so what happens, or what we've realized over the research over the past 20 years, is that cells go into and out of periods where this structure, the way of taking the genome and organizing it into the many different conformations or the many different epigenomes, go through periods of sensitivity. And so this concept is exactly the same as the one first proposed by the pediatrician Maria Montessori. And so she is a very famous pediatrician who noticed that children go in and out of different phases with respect to the development of their visual system, of their motor system, and as well as in the cognition. And that once you are in this period, you have this kind of opportunity to take advantage of that growth, but once you come out of that period, the benefits kind of become less prominent. And so as an example, she made the observation that if you use brightly colored toys with different shapes, that this then pushes forward a stronger interaction between your development of your visual system and cognition, and then once you're outside this window, that doesn't quite work as well. And so, of course, now today she has a whole series of educational interventions in schools named after her, and so the concept here really comes back down to windows. And so just like this, the processes of epigenetic programming also have these windows that they go into and out of, which are going to have maximal impacts. Okay, so very early in development, this window is wide open, and the processes controlling the organization of information in the genome is very plastic, and it has a lot of inputs. However, as we start to age, this process becomes less malleable, less able to be changed, and it isn't until you get into adulthood that this window completely closes, or at least that's what we thought. And so this is why we had this intense interest in understanding early pregnancy and intense focus on mom. However, over the last few years, we started to recognize through various epidemiological studies that male exposures also have these lasting impacts on offspring. And so this paper that I'm putting up here now is one that examined the nutrition of the paternal grandfather, and then how his nutritional status had an impact on the all-cause mortality and cancer development in his grandsons. And so the work that was really initiated here has led to instead of the developmental origins of adult disease hypothesis, or the DOHAD, we now have the POHAD, the paternal origins of adult disease hypothesis. And so what this work has really demonstrated is that males also go through various periods of sensitivity with respect to their reproductive systems. And so there's work been done looking at various inputs or environmental toxins on what happens to germ cell development during pregnancy. And so my colleague, Dr. Sarkar from Rutgers, has done some work here with alcohol. And then once you get out of this period here, there's this slow growth period right before puberty. And then of course, puberty has these windows that are very, very sensitive to alterations in developmental programming. But then there's also starting to be a lot more work looking at adult exposures and trying to understand how these then also transmit memories onto their offspring. And so this is where my work has largely focused. But this area here is very poorly studied. We have no idea whether the changes that are induced at any one of these phases here is permanent, whether interventions like diet or exercise can do anything to push back against these effects. And so this is an area of biology that is still wide open. Okay, and so sperm carry a large amount of epigenetic information that can come in different forms, and so there's DNA methylation, which is the one that's been known and studied the longest. And then there's also a series of what we call chromatin modifications, which are essentially bookmarks that hold different portions of the genome open and then contribute to the organization of chromatin structure in the embryo. And so we do have some reasonably good data to support the role of these epigenetic mechanisms in transmitting paternal effects to offspring. However, the large majority of work has been done looking at non-coding RNAs. And so these non-coding RNAs are small, they don't encode for any proteins, and they get packaged into sperm as a normal part of our reproductive physiology. And so the way that these non-coding RNAs work is very analogous to a cytokine or a hormone. They're just a signaling messenger, okay? And so they come through, and what they're going to do in that early phase of development is they're going to take the thermostat and they're going to reset it at a different level. They're going to basically recalibrate a new normal, okay? And so this can have impacts on mitochondrial function, impacts on growth and development, a whole host of different things. And so the important thing here to recognize is that these non-coding RNAs actually shift in response to various paternal exposures, including diet, various toxicants, exercise, as well as to drugs of abuse. And so the portion of the male reproductive tract that is responsible for programming these effects is this structure here called the epididymis. Okay, and so the epididymis is this tube-like structure that sits on top of the testicle. And so what happens here is that sperm are made in the testicle, they come out of the testicle and swim through the epididymis on their way to the vas deferens where they're stored before ejaculation. Okay, and so as the sperm swim through this tube or this little tunnel, the cells on the side here of the epididymis secrete these little vesicles or these little bubbles. And the bubbles are going to carry these non-coding RNAs over to the sperm and allow them to be packaged into the sperm head and then delivered into the developing embryo. And so effectively what's happening here is that as the sperm are swimming through the epididymis and maturing, the cells here are giving them one last piece of fatherly molecular advice, one more pep talk before they go on to fertilize the oocyte. And what the research from my lab as well as many other labs has shown here is that these non-coding RNAs that are packaged into sperm have a profound influence on the growth and development of the placenta. And so the placenta is the organ of exchange that exists between the mom and the fetus, and it's connected to the baby through this tube-like structure called the umbilical cord, and so this is where we get our belly button. And the placenta has just a fundamental role in controlling growth and development of the fetus. There's a now described placenta brain axis that controls the development of the central nervous system. And importantly, the placenta has an absolutely profound influence on metabolism for the rest of your life. And so the work that we have done, as well as some others, has shown that these non-coding RNAs, we think, are packaged into sperm as a means to help the offspring adapt to whatever environment it is. And so they change the histological organization of the placenta to either favor storage of certain elements or to be more shifted towards absorption. And I'm going to come back and talk about this in just a second. Okay, so just to summarize where we've been here, sperm carry epigenetic information that's going to change in response to male nutrition, lifestyle choices, and toxicant exposures. And epigenetic information carried in sperm alters placental development, and this is going to have lasting impacts on health and disease. Okay. And so now we're going to come to the role in FASDs. And so one of the lingering questions that has plagued this field is why is there so much variation in FASDs? Why is it that some women can drink during pregnancy and effectively get away with it, whereas other women do not? And so this study that I'm putting up here is one that looked at the variability seen in dizygotic twins. And so I think this right here really effectively shows that maternal alcohol exposure is not the sole driver of this condition, that there's other unrecognized factors. And so this leads us to the question, like in other elements of toxicology, is dad the missing piece? And so I like this character here. He is a satirical representation of the American dad, and he's a drinker, right? And so, you know, he portrays beer as being this wonderful thing that he's always chasing after. And so this leads very logically to the question, well, could male alcohol use be contributing to fetal alcohol spectrum disorders? And so this idea that I'm proposing here is certainly not new. There were a number of studies. This one here came out in 1987, and this is the work of Little and Singh. And then, of course, following this work, Professor Abel at Wayne State University did an amazing amount of work looking at this effect in both mice and in rats. However, we didn't understand, or we didn't know about epigenetic mechanisms of inheritance. And so whenever they did this work, they kind of came to this dead end where they said the mechanisms here are obscure, and so we really have no idea how this could be happening. And so this was kind of a dead end. They didn't really know what to go look at after the fact. And so this line of thought was kind of dropped. Okay, and so there's a lot of more work that's been done in looking at male alcohol exposures, and so I can't possibly touch upon it all. And so I just really direct you to this, this very comprehensive review article, which was published by the Humanics group. Okay, and so here we come to the question of where paternal alcohol use fits into reality. And so I want you to please understand that I'm not suggesting that maternal alcohol use is not relevant. What I'm saying here is that I don't think that we have the whole picture in understanding all the different variables that could lead to this outcome. And so my interest in this area of research began one afternoon when I was talking to an FASD researcher by the name of Tim Cudd. And so he was a fetal physiologist who had a very, very intense interest in understanding the impacts on placentation and on early growth. And he described the placental phenotypes to me, and I immediately knew that there was a paternal effect buried in here. And so I had done my Ph.D. studies and postdoctoral research looking at epigenetic abnormalities seen in children conceived through in vitro fertilization or in cloning, animal cloning, and my area of specialty was on the role of imprinted genes. And so many of the changes that were observed here were very similar to what we had described or seen with loss of or alterations in paternal imprinting. And so when I stepped onto this project here, I just had a very simple question. Can paternal drinking induce these placental phenotypes? And is it possible that epigenetic mechanisms could contribute to the penetrance and severity of these effects? And so here are the symptoms of fetal alcohol syndrome, and I don't need to read them off to you because you people all know them probably better than I do. But because I was more familiar with the placental defects, this is where I began my journey. All right, and so to address this question, I used an animal model. So I went to the literature and stole the drinking in the dark paradigm. And so effectively what happens here is that you give the animals alcohol for a 4-hour window during the dark cycle, and so this then encourages the development of binge patterns of alcohol consumption. And so we would expose males to alcohol for about 6 weeks. And so this is going to be in a mouse 2 complete spermatogenic cycles. And then once we have undergone that exposure, we're going to breed them to naive dams. And so in these first experiments I'm showing you here, the females did not see a drop of alcohol. And so the males were kept in a separate cage, and they were only put with the females during mating. And the concentrations of alcohol use that we used here, we actually used a dose response curve. So we had low, which was 3%, medium 6%, and then our high exposure was a 10% exposure. And then the blood alcohol levels that we saw here were in the range of 30 to 160 milligrams per deciliter. And so just for reference, this is about half or a little less than half the legal limit, up to about twice the legal limit. All right, And so just as a point here, the model that we use is one, it's a chronic exposure, so these males are drinking every day, and so this is relevant to about 8.3% of all males in the United States, especially those who are of reproductive age, who are going to consume about 2 to 10 drinks per day. Okay, and so what we did here was to sacrifice pregnancies at gestational day 16.5, and the reason that we did this is because this is where, during fetal development, the fetus is going to turn on the jets and increase its fetal weight, where the most biomass or the largest amount of growth is going to occur during pregnancy. And so if you have a problem with your placenta, this is where you're going to start to see it. And so one of the things that we consistently see here is a placental overgrowth phenotype, where the placentas of the alcohol-exposed males are bigger. And so when we started to do the histology of it, and so here's the map of the mouse placenta. And so the layers that I want to draw your attention to here, you have the decidua, which is the maternal component, and the junctional zone, which is the endocrine component and the glycogen or the sugar storage component of the placenta. And then on top of that, or sorry, below that, you have the labyrinth layer, which is the vascular layer. And so this is where all the blood vessels are going to interface with mom, and this is where the exchange is going to occur. And so what we would consistently see in our model is these dose-dependent shifts where we would see a decrease in the maternal component of the placenta and the endocrine component of the placenta, and an expansion of the vascular layer. And so this is a classic stress response phenotype. So the placenta basically has one move. It can either decrease or increase the size of surface area it has available for absorption. I mean, that's its job. It's this organ of exchange. And so what we see here in this model is that the placenta is stressed and it's increasing its size in order to compensate for some kind of deficiency. And so we think that this placental deficiency actually manifests much earlier in development, because when we do in vitro fertilization, and that is to say we take alcohol-exposed sperm, we do the fertilization in a dish and then get the embryos and transfer them into the recipient dams, we see this dose-dependent decrease in pregnancy success rates. And so we can get an equivalent number of embryos, transfer those embryos into the recipient dams. However, we don't see the same pregnancy success rates. And so one of the larger take-home messages from today is that paternal health is significantly important to IVF outcomes. And then one of the easiest things that males can do to increase their fertility is to stop drinking. Okay, and so like many other studies looking at compromised placentation and compromised growth, we saw that the growth deficiency that occurred during fetal development persisted into postnatal life, and then consistently with other studies in humans as well, that these abnormalities in growth associated with metabolic problems later in life. And so specifically we saw lower fasting blood glucose levels in the males and higher in the females. However, this was during adolescence. When you looked again in adult life, the paradigm had shifted to be kind of more hyperglycemia or high blood sugar phenotype. Okay, so we had, at this point, I think demonstrated to my satisfaction anyway, that we could see placental defects and postnatal growth and metabolic phenotypes in the offspring. And so now we wanted to understand what was the potential interaction here with maternal exposures. And so since we knew that this placental phenotype coming through from the male caused a stress, I became really curious to understand how it is or how it would be that maternal exposures might push this system and what the outcomes might be. Would the phenotypes be equivalent, or would we see some kind of additive or possibly even synergistic interaction? And so this is the direction I've now moved my entire lab in, trying to understand what the interaction is and to see if there is some kind of propensity for male alcohol exposures to interact with maternal exposures and make things get worse. And so to do this, I implemented a mouse model that used a 2x2 factorial design. So we're looking at the offspring of control animals who only ever drink water, what happens when mom drinks, what happens when dad drinks, and then what happens when both parents drink. Okay, and so the model is essentially the same as the one I've just described to you before. We had the males, we exposed them in this case to 10% alcohol, and we had them drinking for their 6-week preconception period. Then for the females, we started them drinking about 7 to 3 days before mating, and then we continued those alcohol exposures through to gestational day 10. And so we did this because we suspected that if we wanted to model the circumstance where a mom found out she was pregnant but then stopped consuming alcohol. And so when we did these experiments, the blood alcohol levels that we saw were right around the legal limit here. So we saw 80 or 92 milligrams per deciliter, which again is kind of right around that, the legal limit to drive here in the United States. All right, and so in this series of experiments I'm going to show, we really took an interest in understanding what the impact would be on craniofacial phenotypes. And so to do this, we stole the work of this group here, Klingenberg et al., who published this study in alcohol using this technique called geometric morphometrics, which is a digital technique that is going to analyze a picture of the face. And so effectively what happens here is you take the face, you calculate the size of the eyes, the angle of the eyes relative to the nose, to the mouth, et cetera, and then all of this information gets put into a matrix. And so as I was walking through the airport on Thursday, the Department of Homeland Security was looking at my face to see if the mathematical relationship of my eyes to my nose to my mouth match any bad actors. And so effectively, this is the technology by which facial recognition occurs. It's also a variant of this is what Google and Apple use to unlock your phone. Okay, so what we did was to take mugshots of the developing fetus, and then we had an undergrad who would sit there for hours and hours putting on these various landmarks. Okay, so you demarcate the tip of the nose, the top of the head, so on and so forth. And then you take these data and you remove scale by putting them into a matrix, and then once you've done that, then you can start doing some advanced statistical modeling. And so what we saw was that maternal alcohol exposures pushed the population in one direction, paternal alcohol exposures pushed them in a slightly different direction, but then the dual parental exposures kind of pushed them in their own direction. So each one of these different exposure models had its kind of own unique effect in terms of which specific subsection of the face was impacted. But for our experiments, the outcomes here matched very, very nicely some of the work that was done in that human study, where we saw that the vast majority of changes occurred in the lower aspects of the face, particularly in the jaw and the positioning of the ear. And so we observed micrognathia and retrognathia. And so I've just, as at this meeting, made some dentist friends, and so I'm very excited to set up that collaboration and explore the impacts of alcohol exposure on this phenotype. Okay, and so just like in human studies and in mouse studies, we see that paternal alcohol exposures also have these dose-dependent effects on offspring brain weight, and that this can be slightly exacerbated in the dual parental exposures where the slope of the line is actually a little bit more severe. We haven't done the hard statistics on this to figure out if that actually is going to emerge and be a full result, but we're still working on figuring out how exactly to model the dose of the mom and the dose of the dad combined. But just like in human studies and some of the talks that we've seen here, we see an impact on head size or micro, is that right? No. Microcephaly, yes, there we go. Microcephaly, and it doesn't matter whether it's paternal, maternal, or both parents, that particular phenotype is there. And so from a paternal point of view, we can see in this mouse model all of the various phenotypes that emerge in maternal models of exposure. All right, so to summarize these last points here, using a mouse model of chronic male alcohol exposure, we can see the core FASD symptoms. And maternal and paternal alcohol use interact in driving some of these related phenotypes. I'm going to come back and talk about this a little bit more towards the end of this section. Okay, so the question here is how? How is this memory transmitting across from dad to offspring? So there is a massive disconnect here between the exposure when it's actually occurring, particularly with our IVF studies, where the males had been killed and their sperm collected 6 to 8 months before fertilization even occurred. So there's this epigenetic information that is somehow being perceived by the dad and then programmed into the sperm, and then that information is going on into affect the offspring as yet through some identified mechanism. And so the question that I wanted to ask here, or try to understand, is what is the form of this information? And so one of the important things here is that because we are seeing these dose-dependent effects, this isn't going to be a binary signal. It's not going to be a 0 or a 1. There has to be some kind of graded information coming through, which is going to make detection a little bit more complicated. And so this is what I wanted to research and what I'm going to focus the rest of my talk on. And so when I had my job interview to stand up here in front of you, both CJ and Maggie said to me, well, we need to understand why we'd even do this research in the first place. What benefit is it? And so I've developed this slide here to address these important points. Okay, so the first one here is that we need to identify the signal in sperm because it might be present and arise from other exposures. And so we've known for a while now that obese males actually have a stronger impact on their offspring developing metabolic dysfunction than maternal obesity does. And so we need to start understanding how these signals move from dad down to offspring. But another important point here is that we could measure this signal in IVF clinics and warn couples that, hey, the male needs to stop drinking because this signal is popping up and this is a risk for recurrent pregnancy loss. But I think one of the more important things here is that we have to be able to measure this in order to figure out how long it takes for the effects of alcohol to flush out. If males don't know how long to stop drinking before conception, they're going to ignore the problem, right? If there's no hard number here that says stop drinking 3 months before you try having a baby, then it just becomes open season to whatever you think is best. All right, and then finally here, we need to understand what changes are occurring in the offspring, and this would maybe allow us to develop some kind of intervention. Okay, so to begin this journey, I went back to the RNA sequencing data that we had collected for a variety of different tissues in dad, as well as the tissues in his offspring. And so what I'm showing you here, this is the mouse epididymis here. This is the in vitro fertilization embryos we developed using alcohol-exposed sperm. And then these are the placentas that we use from our studies that I just mentioned very briefly, or previously. And in each of these circumstances, the thing that emerged the most prominently was mitochondrial dysfunction. Okay, so this was kind of the recurring theme that kept popping up over and over again in these analyses, to the point that I really lost enthusiasm for doing RNA sequencing ever again because it just comes back with the same answer. Okay, so mitochondria. Our mitochondria are these wonderful little organelles that exist inside our cell. What they're going to do is they're going to take the food we eat and use those components to push protons up an energetic gradient in order to generate electrochemical energy. And so effectively what your mitochondria are your battery. Okay, so this is the battery of the cell. And when you have dysfunction in the battery of your cell, it is going to lead to a large number of different pathologies. Okay, so if you don't have functional mitochondria, you can't make hormones as effectively. You have problems with beta fatty acid oxidation, so you're going to end up with problems with liver function. This will bleed through into other aspects of metabolism, and then you also have relationships here to accelerated aging, inflammation, and cancer. And so this is an area of burgeoning research that we're trying to understand called inflamma-aging, understanding how it is that the immune system and inflammation might be driving forward these processes. And so we've known for a long time that alcohol is brutal to our mitochondria. In fact, some of the first studies ever done using electron microscopy were done looking at liver mitochondria isolated from alcoholics. And so the reason for this is that the metabolism of alcohol leans very heavily on the mitochondria, and some of these products that it generates are ones that can cause the normal mitochondrial function to collapse. And so what we wanted to do here, after seeing this signature emerge from our research, was to go back and try and understand, do we see evidence of mitochondrial dysfunction in our model? And we wanted to pay particular attention to what was going on with mitochondrial complex 1. And so this is that giant lantern battery that I had in the previous slide, and this is the one that's going to be responsible for generating most of the oxidative stress when you have mitochondrial dysfunction. All right, so we went back to the males that we had exposed to that dose response and started to look at the effects of chronic alcohol exposure on their mitochondria. And what we saw was that we saw dose-dependent decreases in various mitochondrial complex members, as well as dose-dependent increases in mitochondrial copy number. And so this last graph right here is an important one, because what we see is that the more the male drinks, and when we go up here we're looking at dose, the more the male drinks, you start to see a higher and higher number of mitochondrial copy number appearing inside his cells. And so I don't want to go into the biochemistry of it here, but there's a pathway called the C-gas sting pathway, which is the pathway by which your mitochondria scream for help and initiate the process of inflammation. Okay, and so now that we had seen that, we went to the males and we isolated the sperm small RNAs from the sperm, and we sequenced them. And so what we saw here was that at the low and the medium dose, we had a dramatic enrichment of microRNA miR200C, and at the medium and high dose, we had a loss of microRNA 465. And in fact, the entire group of microRNAs from this cluster. And so these microRNAs are just an epigenetic signal. The specifics here are not so much important, just that we saw these dose-dependent shifts in these epigenetic signals. And these results here mirror some of the results published by Greg Homanic's group, who has also looked at changes in non-coding RNAs in sperm induced by alcohol. And so this was reassuring to see that we were on the right track. Okay, and so this figure right here kind of represents the various changes that we see. There's some microRNAs that go up with alcohol and stay up. There's some that are lost. But this one here, this microRNA 200C, had a very dynamic pattern and had this prior association with ischemic stroke. And so if you have a stroke event, microRNA miR200C is one of the ones that actually makes things worse. And so if you develop a therapy to intervene against this microRNA, you can actually lessen the effects of stroke. All right, so then this led us to our working hypothesis that the mitochondrial stress caused by alcohol in dad is going to transmit a memory to his offspring, potentially adversely affecting the mitochondrial function in his kids. And so what we call this is a adverse or a maladaptive form of mitochondrial adaptation. And so this research here, this convergence of this research here with the stroke literature really pointed us towards understanding the impacts on mitochondrial complex 1. And this is because this event of stroke and the involvement of miR200C really has these adverse effects on ischemic stroke, and then has all kinds of relationships to accelerated aging and the development of age-related diseases. And so what I did here in this next series of experiments was to examine mitochondrial function. And so to do this, I got out my handy-dandy voltmeter, and I focused in on the bigger part of the battery here, complex 1. And so I'm going to show you data here from the gestational day 16.5 brain, as well as the adolescent liver, okay? All right, and so in this assay, effectively what we do is we turn on the car, we hook up the voltmeter, and we see how the battery handles the stress. And so when we do this experiment, what we see here during fetal development is that the normal function of the battery is going to go on this straight line, whereas during in the offspring of alcohol-exposed males, you have this diminished capacity, which is much lower than the offspring of unexposed males. And then similarly here, when we go and look at the mitochondrial function in the adolescent liver, now we really see that these phenotypes here are starting to pull away. Okay, and so the trajectory of this line is not the same as the trajectory of this line, so we're really starting to identify pretty significant deficits in mitochondrial function. All right, and so adversity or problems in mitochondrial function are going to have bleed-through effects at multiple different stages of your life. And so as an example here, bad mitochondria are going to significantly impact the process of differentiation. So as an example, the neural crest cells that migrate to give the face, they have to undergo this mitochondrial transition in order to let go of their neighboring cells and undergo this, what we call the epithelial to mesenchymal transition. Similarly, even in adult tissues, there's a mitochondrial flux or a change that needs to happen during pregnancy in the breast, and so the mammary gland has to undergo this mitochondrial transition in order to get prepared for lactation, and I have a colleague who studies this process within the context of understanding breast cancer. And then similarly here, if you don't have well-functioning mitochondria, you can't drive forward basic metabolic processes, including the breakdown and function or breakdown and processing of fats. And then as I talked about very briefly before, you also have this direct interaction between mitochondria and the processes controlling inflammation. And so Joanne Weinberg's group has previously talked to you, I'm sure, about the role of alcohol exposures, prenatal alcohol exposures in driving immune dysfunction. And so effectively we're seeing the same thing here in the offspring of alcohol-exposed males. And so in this next experiment, I'm going to show what we did here was to look at the secretion of this inflammatory cytokine, IL-6, excuse me, IL-6 across the life course in the offspring. And so here, I'm not just looking at the offspring of alcohol-exposed males. I've got mom in red, dad in blue, and then both parents here in gray. And what we see is that at adolescence, that it's the dual parental offspring of dual parental exposures that show this hyperinflammatory phenotype, the highest secretion of IL-6, whereas maternal and paternal alcohol exposures alone do not. However, once you get into adult life, you can see here that there's this additive effect where, yes, both maternal and paternal alcohol exposures are driving this increased secretion of this inflammatory cytokine, but over here in the dual exposed offspring, it's already higher than it is at this stage. And then by the time you get into old age here, so here we are at about almost a year of age for these mice, the effects have kind of maxed out. And so the inflammatory phenotype for the dual exposed offspring has been present for their entire life, whereas this high level of inflammatory cytokines is really only present adult stages and onward for the singly exposed offspring. And so just as a minor point here, I was very moved by Carl Jung's talk and the relationship or the experience he had with fibrosis. And so this cytokine here, IL-6, is a major driver of liver pathology, including fibrosis. And so it gratified me to hear that something I'm doing is on the right track. Okay. And so the ongoing experiments that we have right now are trying to understand if we can induce these phenotypes by injecting some of the microRNAs that we've identified in sperm. And so to this effect, I collaborated with Colin Conine at the University of Pennsylvania, who is a hardcore RNA biologist and embryologist, and so he's done the injection experiments to introduce miR200C at a physiologically relevant level. And the preliminary data that we have on this from RNA sequencing of the injected embryos shows that we're activating the EIF2 stress pathway here, which is indicative of an unbalanced electron transport chain. So we think that we're on the right track here, and so we're doing the histological analysis on the placenta hopefully as we speak. All right, so to summarize these last points here, so chronic alcohol exposure causes dose-dependent increases in mitochondrial stress and epigenetic signals in sperm, and this male alcohol use transmits a memory that alters mitochondrial function during fetal development and persists into later life. All right, and so to kind of reemphasize the plausibility of this, I want to just point out here that the idea that parental exposures, both mom and dad, have an impact on offspring mitochondrial function is not new. Biologists who have been studying lower animals like worms have been studying this phenomena for years. And so research in these species here have really shown that the transmission of this effect is tied to the NRF2 protein, which is a big component of the antioxidant pathway. And so I had the NRF2 null males, or the gene knockout males, in my lab. And so the experiment that we put together is this one right here. I took the males who are knockout and bred them to wild type females. And then we looked at the placental phenotypes and growth phenotypes in their heterozygous offspring. And so this right here goes into some technical jargon, so I'm going to just take a minute here and go through exactly what it is that I've done and what it means. Okay, so this right here is your mitochondria, and on any given day, your mitochondria are going to start to produce what we call reactive oxygen species, and so long as that level is a low level of production, things are fine. However, if you have a Coke or you do something that otherwise stresses your system, well, then you're going to start to produce an excess of these reactive oxygen species, and they're going to go throughout the cell and wreak all kinds of havoc. And so in this example here, we've spread that reactive oxygen species over here to the ribosome, the thing that we use to generate new proteins, and this is going to have an adverse effect on cellular physiology and function. And so to help clean up the mess, we have a hazmat guy, okay? That's Mr. NRF2 here. And so Mr. NRF2 comes into the situation, and he has the expertise and the equipment to gather up those reactive oxygen species and to dispose of them safely. All right, and so what happens here, what we did, and I say the royal we, I didn't do this, this is something that, an existing resource that was available, they went in and they excised, or they cut out the gene for NRF2. And so because you don't have the information for NRF2, you don't have the hazmat guy, and you have this chronic state of oxidative stress. So this mouse is effectively hung over all the time. It's not a perfect analogy, but that's kind of the gist of it. This mouse is always going to be experiencing oxidative stress. And so this mouse has a predisposition to develop liver disease, it ages faster, and has a large number of other health and fertility issues associated with oxidative stress. All right, so then once we sequenced the sperm from these NRF2 males, what we noticed was the same signature was there. So we saw an upregulation of miR200C and a loss of miR465. And when we went and started looking in their offspring, and so just as a second here, I'm going to kind of explain this to the other scientists in the room. NRF2 is a stress response protein, and so it's held in the cytoplasm when it's not being used, and when it's held in the cytoplasm, it gets ubiquitinated, taken over to the proteasome, and destroyed. And it isn't until those reactive oxygen species show up, free NRF2 from its keep1 protein, the protein that's anchoring it in the cytoplasm, that it's allowed to translocate into the nucleus. And so what we see in the offspring placenta here, so this is the heterozygous offspring placenta, is very similar to what's been reported for other stress response proteins that have heterozygosity. And so you have this no difference here in the level of the protein between the wild type animals and the heterozygous offspring. And so from this, we infer that any of the effects that we are seeing are due to these paternal epigenetic changes, not differences in the level of the NRF protein per se. And so what we see in the offspring is, again, you have this decline in the component of the placenta here responsible for storage and the interface with mom, okay, the decidua, and you have this increase here in the labyrinth or the vascular layer of the placenta. And so interestingly here, we see this increased labyrinth layer in both the male and the female offspring. And so we think that this is because the phenotype is consistent and significant enough that it's able to push the phenotypes of the females towards this condition. All right, and then finally here, we did the analysis using that software, that Morpho-J software, the analysis of craniofacial shape. And again, we see these changes in the shape of the face that predominantly localize to the lower aspects of the face, including the jaw and the positioning of the ear. All right, so to summarize this last point here, male oxidative stress by itself can alter epigenetic signals in sperm, which are going to impact placental health and craniofacial shape. All right, so now we've filled in a few more blank spots in this model here. We think that paternal mitochondrial stress or oxidative stress is a signal. We don't know if it's being measured in the liver and then transmitted down to the reproductive tract, or if perhaps the cells of the reproductive tract can measure that independently. This information is being put into sperm. It jumps across here to the embryo and then alters mitochondrial function in the offspring with these long-lasting effects that persist through fetal development and at least into adolescent life. And so we've long known that alcohol exposure is bad for mitochondria, and so I think that from our research that it isn't going to be an acute response that's responsible for driving these effects, but nonetheless there's a group of people out there for whom a drink is not a drink, right? So this is well, I don't need to explain this to this audience. But there are other things, certainly in our environment, that modify mitochondrial function, okay? The American diet is absolutely brutal to mitochondrial function, and a sedentary lifestyle is also not good for mitochondrial health. So we have a number of different things that we're exposed to in our school systems or in our day-to-day lives that we know are not good for us, and we know, impact mitochondrial health. But then, of course, there's the other things that we can't see, that are toxins in our environment that are there, put there by our parents and our grandparents, the endocrine disruptors, which most of us are blissfully unaware. But these chemicals, they also have an impact on mitochondrial function. And then finally, there are the stressors that we do to ourselves because we're bored, right? And at the same time, profoundly lonely. And so here it is, we look at our screens and we doom scroll. We see what Trump has said. We see about the war in the Ukraine. We do all these things and our cells are listening, right? The mitochondria are listening, trying to figure out how they can help us adapt. And each of these toxicants, each of these exposures that I've talked about today, all of them impact mitochondria. And so our mitochondria have enabled humanity to live above the Arctic Circle, into the desert, to survive and thrive on a wide range of different diets. And so it's not surprising that these little organelles, which have helped us survive natural disasters, plagues, so on and so forth, have the capacity to speak to our germline and to have this impact on lifelong function and health, because that's their job. They're helping us adapt. So we need to understand what these signals are that allow mitochondria from the parent to communicate to the offspring so that we can understand the full range of factors that are responsible for driving pediatric disease and understand fully the environmental determinants of lifelong health. I like to use art rather than scientific studies because it's just a little bit more impactful and easy to understand. All right, and so with that, I'd like to thank the people who gave me the money to do this, including the Keck Foundation, as well as the NIAAA. And of course, I didn't do any of this research. I just stood back and let these excellent people here do what they do best. And so with that, I'd be happy to take any questions.
I too would like to thank Dr. Golding for a wonderful talk. We have time for about 3 questions, and so there is a microphone there, and I see Raj already there, and then Joe has agreed to run around with a mobile phone, a microphone as well. And just to remind everybody that there will be slides posted on the website 1 week after the conference. So if you were busy taking photographs and missed some, they will be available. And so just to remind you all of that. And so, Raj?
Thank you for probably one of the most amazing Sunday morning talks that I've been to for a long time in terms of paradigm shift of how we think. Amazing, so thank you. You kind of alluded to the question in your talk, is if we now go and point this out to people, because we will be going out and teaching, and you will get the question is, what's the paternal contribution? And we can't say there isn't one now, because you've just shown there is. The question we're going to get asked as clinicians on the ground is, how long do we have to not drink beforehand?
Yeah.
Because sperm are being produced all the time.
Right.
And that is the fundamental question I guarantee you we will be asked. And what do we know about that? Because you didn't actually answer it, even though you raised the question.
Yeah, so this is a problem, and so it extends not only from the consideration of alcohol, but into reproductive biology in general. So I'll just kind of quickly summarize this. When you have an infertile man, the treatment for the infertile man is to treat the woman, right? You superovulate her, you make her go through in vitro fertilization. And until we start paying attention to male reproductive health, we don't know. Like, we don't even, the things that we're measuring today are the same things we were measuring in the 1930s. And that's ridiculous, right? So we have to expand, the things that we measure, and clinicians might know this, but the general population might not. Sperm samples are actually not that easy to collect, or that difficult to collect. So it should be something that we should be measuring and able to measure, fairly routinely, right?
Thank you. He said already my question, but now I have just a suggestion. Since we are here, I hope for future research also we cover the impact of maternal alcohol consumption before pregnancy, not just during pregnancy, because I read before some articles talking about DNA can be altered by alcohol on the egg and also the sperm. And also other research, if we can talk about how the impact of maternal alcohol consumption after birth, during the lactation or the breastfeeding on the baby. Is it safe to use it or not? And if they can't use it, for how long? All this information, I hope that next years we can put it in our research. Thank you.
Absolutely. It is hard to study maternal exposures, especially preconception ones, because you don't know if the effect is a lingering effect, whereas the male exposure is much cleaner. Yeah.
Thank you. I think it was an amazing talk. Just a comment for the audience, and the probably first description of what we know as fetal alcohol spectrum disorders, which was made by Paul Lemoine, a French pediatrician, in 1968. The article is called "The Children of Alcoholic Parents," and the demographic data and explains about the use of alcohol in father and mother,
right,
just as much.
Yeah, that's a fantastic point. Yeah.
Okay.
We're good?
Yeah, I think everybody is ready for coffee and a break. So again, let us give Dr. Golding thanks for a wonderful talk.